Now, in the part 2, if we are going to talk about the human immune system, two keywords should be known, that is antigen, which is a foreign particle come into our body and may be harmful or may not be harmful, but as it is not part of our body, it should be removed. For that, antibody is produced. So, on the basis of these two things, the total human immune system is basically based. So, human immune system is the system that gives immunity to the body so that we can recognize, responding, and remembering the foreign pathogen. It plays role in allergic reaction, autoimmune disease where antibody cannot identify whether it is the part of the body or the outside of the body, and during organ transplantation. So what are the organs included in the human immune system? Lymphoid organs, tissues, cells, and antibodies. Now, if we are going to talk about the lymphoid organs, so these are the organs where origin, maturation, and proliferation of lymphocyte occurs. So, we have two types of lymphoid organs, primary lymphoid organ and secondary lymphoid organ. In the primary lymphoid organs, the lymphocyte are matured and differentiated to antigen sensitive lymphocyte, and these are of two types, bone marrow and thymus. Now, in the bone marrow, the formation of blood cell like RBC, WBC and platelet are formed, and also B and T lymphocyte are also formed. Now, thymus, it is a bilobed organ you can see in the picture, which is seen near the heart and beneath the breastbone. At the time of birth, it is large, but as we are going to the puberty, it gradually reduces, and at the time of puberty, it becomes very small in size. Now, immature T lymphocyte, when they are originated from bone marrow, then they migrated to thymus, and there the T lymphocyte become matured. Now we are going to talk about the secondary lymphoid organs, the organs where the matured lymphocyte come after the maturation from primary lymphoid organ. If we say very simply, in the primary lymphoid organ, the lymphocyte originated, matured, differentiated, and then migrate to the secondary lymphoid organ. And and then they interact with antigen and then proliferate to become effector cell. Example, spleen and lymph node, tonsil, Peyer's patches, mucosa associated lymphoid tissue in the short form MALT, and appendix. So these 6 organs are the secondary lymphoid organs. Again I am saying, which are the secondary lymphoid organs? Spleen, lymph node, tonsil, Peyer's patches, mucosa associated lymphoid tissue, and appendix. Now the spleen, it is a bin-shaped organ and it contains lymphocyte and phagocyte. It removes worn-out RBCs and also microorganisms from blood. Basically, it is the reservoir of erythrocyte, means RBC, in the fetus. Now, if we are going to talk about the lymph node found in the lymphatic system, they trap microorganism or other antigen. Trapped antigens activate lymphocyte and cause the immune response. Now the MALT, which is the mucosa associated lymphoid tissue, basically it is located within the lining of the respiratory, digestive, urinogenital tract, and it constitutes 50% of the lymphoid tissue. Now if we are going to talk about the immunity, then it is the ability of the immune system to fight the disease-causing organism. See, we always exposed to different kind of pathogens, but we are not affected to every organism. Why? Because our immunity to some extent able to fight with some microorganism, and our immunity cannot be able to fight some kind of microorganism. If it can able to fight, then that microorganism will not be affected will not affected us. But if the immune system become looser, then we feel sick for that pathogen. So now the types of immunity are 2. One is innate immunity, by which we are born. You can say it is a God-gifted. And another is acquired immunity. Throughout our life, we acquired this immunity. Now, innate immunity, it is very non-specific defense. It not see that which kind of pathogen is entering. They are just going to destroy any kind of foreign particle, and it is present at the time of birth. So basically it provides the barrier to the entry of foreign antigens into our body. So these barriers are basically 4 types. Physical barrier, physiological barrier, cellular barrier, cytokine barriers. So physical barrier, which is also known as First line of defense, skin prevent the entry of foreign bodies, because our whole body is covered by the skin and skin is impervious for the foreign particle. So it prevents the entry. Mucosal coating, it creates the lining of respiratory, gastrointestinal and urinogenital tract so that it can trap the microbes. Now, if we are going to talk about the physiological barrier, they prevent the microbial growth, includes gastric HCl, saliva, tears, etc. They have some kind of enzyme present in this secretion and which digests the antigen. Now the cellular barriers, phagocyte kind of cell like WBC, example neutrophil or polymorphonuclear leukocyte, monocyte and natural killer lymphocyte, macrophage, they, like the amoeba, create the pseudopodia, entrap the antigen and digest it. Now we are coming to the cytokine barrier. Virus infected cell secrete a cytokine protein named as interferon. So it protects the non-infected cell from further viral infection. So cytokine basically made for particularly virus. Now acquired immunity. This immunity is very much specific for the pathogen and it is developed during lifetime. It is characterized by memory, that is, during first encounter of a pathogen, body produces primary response which has a low intensity. So the pathogen can create a different kind of symptom. But when the second time the same pathogen wants to enter, we have a memory cell. So the secondary response basically in high intensity. Now the primary and the secondary immune response are carried out with B lymphocyte, that is B cell, and T lymphocyte, that is T cell. You can see the B lymphocyte who can produce antibodies. These are the proteins to fight with pathogens, and T lymphocyte help B cells to produce antibodies. Now we know that B cell can produce antibody. So how the antibody is look like? An antibody has 4 polypeptide chain, 2 light chain, that black and white, and 2 heavy chain, that black and sky blue. That why its formula is H2 for heavy chain, L2 for light chain. There are different types of antibody: IgA, IgG, IgM, IgE and IgD. Now acquired immunity are basically 2 types: humoral or antibody mediated response. Second is cell mediated response or cell mediated immunity. If humoral or antibody immunity response. It is the immune response which is mediated by the antibodies. Now these antibodies are found in the blood plasma. So it is known as humoral immune response. Now the cell mediated, it is the immune response mediated by the T lymphocyte. The body can differentiate self and non-self, and the CMI, same cell-mediated immune response, causes graft rejection. Now, tissue matching and blood group matching are very much essential before undertaking any graft or transplant. After this, the patient has to take immunosuppressant all his life so that antibody cannot reject that thing. First of all, we have to match the donor with the patient. If we see the blood donor, if the recipient is O, the blood donor should be O. If the recipient is A, blood donor can be O and A. If the recipient is B, blood donor can be O and B. If the recipient is AB, blood donor can be O, A, B and AB. Now, the types of acquired immunity, basically two. One is active, second is passive. Active are of again two types, natural and artificial. Passive is also are of two types, natural and artificial. Now if we talk about the active immunity, it is the immunity in which antibodies are produced in a host body when the host is exposed to antigen. That means maybe it is living or dead microbes or other protein. If injected into the host body, then the body creates the immunity. It is a slow process. It is produced by two ways. Natural active immunity means it is developed during natural infection by the microbes. That microbes enter the body and create the infection. And artificial means it is developed by injecting the microbes deliberately as a vaccine so that immunity can be developed. Now if you talk about the passive immunity, here ready-made antibodies are directly given to the body. It is of two types, natural, passive immunity. The examples are antibodies IgG from mother to placenta to fetus. Now antibodies which is present in the breast milk called colostrum to the infant, and artificial passive immunity anti-tetanus serum. Now immunization. This is based on memory of the immune system. One is active immunization, two is passive immunization. Active immunization means vaccination. In this, a preparation of vaccine means antigenic protein of pathogen or inactivated pathogen is introduced into the body. It results in the development of the antibodies. So during actual infection, the antibodies neutralize antigen. Now, the vaccines also generate memory B and T cell. They recognizes the pathogen quickly Example, polio vaccine, hepatitis V vaccine, DPT vaccine, etc. Vaccines are produced using DNA recombinant technology. Example, hepatitis V vaccine produced from yeast. Now, it is the passive immunization is the direct injection of preformed antibodies or antitoxin. It is required for quick immune response. Example, immunization against tetanus, snake venom, etc. Now, allergy. It is the exaggerated response of immune system to some antigen seen in the environment. Allergen, substances causing allergy. Example, mites in dust, pollen, animal dander, fur, etc. So antibodies produced against the allergen are basically IgE type. IgE binds on mast cell so that it can release the chemical like histamine and serotonin from them. It results in allergic reaction. Now, treatment drugs like antihistamine, adrenaline, steroids quickly reduce the symptoms of allergy. Asthma is a respiratory disease due to allergy. Modern day lifestyle results lowering of immunity and more sensitivity to allergen. Many children in metro cities suffer from allergies and asthma due to sensitivity to the environment. This is due to the protected environment provided early in life. What is autoimmunity? In higher vertebrates, memory-based acquired immunity evolved based on the ability to differentiate foreign organism from the same cell. It is the normal immune process. Sometimes due to genetic disorder or any other unknown reason, body cannot recognize what is the foreign antigen, what is the self antigen. So body attacks self cell. As a result, body become damaged. This is known as autoimmune disease. Example, rheumatoid arthritis. Now we are going to see the third part of this chapter.